Thanks a lot, Brian, and thanks to Brian and Eric for organizing the retreat and for inviting me. I'm honored to be here and to be invited by my peers. And I thought the poster session last night was fantastic. I was very impressed by the quality of the presentations by the students and also the lively discussion that we had there. A little bit of background on me. So I'm relatively new to Georgia Tech. I've been here about a year. I'm delighted to be here and be part of the School of Biology. I was at Florida State University for 12 years before that, and I was in an oceanography department. So you'll see that in what I'm presenting today. What we do is very much a blend between the microbiology and the geosciences. And what I'm going to talk about is part of the work that we're doing in our lab. Half of the work actually in our lab is focused on marine science, and the other half is focused on terrestrial microbiology. But I'm going to talk about this sort of odyssey that we've been on for the last more than 2 years now since the Deepwater Horizon discharge. So I'd like to thank the agencies that have funded this work. It started out funded by the National Science Foundation with a couple of rapid grants to myself and Marcus Hoodle at Florida State University. And currently we've moved out to the deep ocean now. I'm not going to really talk about that today, but that's funded by the Gulf of Mexico Research Initiative, and in particular a consortium called Deep Sea, also at Florida State. So here's what I want to do today in 40 minutes or less. Talk about the economic and ecological value of the Gulf of Mexico, some about the science of the Deepwater Horizon spill, talk about impacts and response, and in particular my own research on that. I wanted to just talk a little bit about what we do in my lab. If we were to explain what we do in one sentence, I would say we study the role of microorganisms in ecosystem function. Now, how do we do that? We do that at the interface between microbiology and geochemistry. So we grow organisms and we study their biology in the lab. We look at their activity using geochemical techniques like this mass spectrometer you see here that we use to look at aqueous gases in marine samples. We use this variety of approaches to look at the mechanisms and controls of biogeochemical cycles, and then through that we try to understand ecosystem function. So I start out when I talk to a broader audience about this by asking questions. And so what I'd like you to do is just take a look at these questions, think about them while I go through my introduction about the Gulf and the Deepwater Horizon, and see if we can answer these questions throughout the talk. The first one is kind of obvious. That is, what dominates the revenue stream of the Gulf states, the 5 Gulf states that ring the northern part of the Gulf of Mexico? What ecosystem services are provided by these ecosystems, and really all, at least coastal ecosystems? Did this Deepwater Horizon accident represent the largest spill in history? And then where did most of that oil go? My neighbors ask me that question all the time. Well, it seems like it wasn't that big of a deal, Joel. Where did the oil go? What's going on? So let's start out talking a little bit about the Gulf. Here you see a map bordered by the 5 states that I mentioned to the north, Mexico to the south, and Cuba also here. It's a relatively small ocean basin. It's only 1,000 miles east-west and roughly 560 miles north and south. It's greater than 2.5 miles deep, and it averages 1 mile deep. So this is a small ocean basin. The Deepwater Horizon accident occurred about here in a mile of water depth. So for such a small ocean basin, it has a huge amount of biodiversity. It was discussed yesterday. Jeanette asked Frank, well, what's your favorite sea experience? And I think that's relevant to here too. One of the amazing things about being involved in this project is you find out how much wild stuff is in the deep Gulf of Mexico. So we're studying microbes all the way up to megafauna, and there are people catching sharks that are bigger than, with heads bigger than my body, Big old snake eels, coming out that are like, 20 feet long. So that's amazing, and something I've also been fascinated by is the deep water corals that are found there. The corals that are found on the West Florida Shelf are actually a lot healthier than the corals that we find in the Keys. And I was talking to Mark Hay a little bit about that. So it's just fascinating to be involved in this and find out, you know, how much wildlife is still down there and relatively untouched by human beings. In order to understand the effects of oil spills on these habitats, we really have to understand how the Gulf works. There's a paucity of information, especially on the deep ocean environment in the Gulf of Mexico. So you can't really see this too well. I wonder if we can turn it down, the lights a little bit, or...
Wait, Mark.
So,
Mark, up there. They're trying to film Joel.
Okay. Okay, so I guess we can't. I put this up here. I actually, this is from the National Geographic. I printed it out and put it on the wall at Cherry Emerson just because I wanted to have something to show people when we talk about these habitats. But just the variety of habitats that we find in the Gulf, from the cold seep environments, which are separated from photosynthetic carbon inputs, to the deep water coral habitats. Of course, fringing marshes and mangroves are a big part of the coast of the Gulf of Mexico. Perhaps most importantly, though, the planktonic habitats, right? The planktonic food web should be driving a lot of life that's in the Gulf. And so impacts of oil on that will be particularly important. So I think this is probably obvious to you, especially the ecologists. What are the services that Gulf habitats and ecosystems provide to human beings? Well, they provide clean water. They provide fish and shellfish. 1/3 of all the fish and shellfish that are eaten in the United States, or seafood that's eaten in the United States, comes out of the Gulf of Mexico. And of course, that's dependent on having clean water and clean ecosystems. If we go one step further and we talk about, well, how is the economy and the ecology of the Gulf built? Well, if we look at the economy, if you look on the left-hand side here, you see that revenue is dominated by oil and gas. That's not a surprise, right? The 5 Gulf states are primarily fueled economically by oil and gas. But if you look on the right-hand side here, you see that the jobs, there's actually 5 times more jobs in tourism than there is in oil and gas. Right? So I use this to make the point that natural resources have to be in balance, right? The resources that we use for oil and gas have to be, or the extraction of those resources has to be balanced with the protection of these very sensitive habitats that are important to tourism, that are important to seafood, and all of the other ecosystem services provided by the Gulf. So to take that one step further and show you that in this slide, you see the schematic of the very sensitive fringing wetlands off the coast of Louisiana. So New Orleans is right up here. You can't see it too well. The green is freshwater marshes, and this blue color here is the salt marshes. And then if you look at the next slide here, you can see that huge numbers of oil and gas rigs shown in red here. I also have this printed up and plotted in posted in Cherry Emerson. These shaded areas are the oil and gas leasing sites. The red dots are all the oil and gas rigs. And all those sensitive marshes that I showed you are right up here, with New Orleans being right here. So we have, again, the sensitive habitat juxtaposed with huge numbers of oil and gas rigs. So I often say that it's not a question of whether there's going to be another spill, but when it will happen. And so here's an example of that. There's still a lot of oil in the Gulf. Something like 9 to 11% of the oil and gas reserves that we have left in the world are in the Gulf of Mexico. If you look at this plot up here, you see that we're drilling deeper and deeper. The oil production is going deeper and deeper. And then in this plot here, you see that the projected reserves, in other words, the deeper you go, the more oil you can extract. Okay, so that's going to be happening in our future. It's already happening. They're drilling test wells off the coast of Cuba. There's a lot of pressure in Congress for us to drill deeper and deeper, so this problem is not going to go away. Okay. Just a few facts about the Deepwater Horizon event in particular. It happened in April 20th. Well, the blowout happened April 20th of 2010. It took 2 days for the rig to burn and then fall to the bottom. 11 people died. The largest part of the tragedy, one could argue. And it's just a few facts. We call it, you might have seen some of these terms in the media, Macondo Prospect, Mississippi Canyon, Block 252. MC 252 just refers to the part of the seafloor that was leased where they were extracting the oil and gas. It took until July for the rig for the leaky pipe to seal, and so that was 86 days that oil was leaking out. A total of 5 million barrels of oil were released into the Gulf of Mexico, and you have to consider too that's should be 1/3 again higher because there was 1/3 of gaseous hydrocarbon release in addition to that liquid hydrocarbon that they often talk about in the media. And another interesting thing is here you see that comes out to about 60,000 barrels a day. Does anybody remember the initial estimates that the Coast Guard was giving per day in barrels? Quarter of that 60,000 barrels? Anybody else have an answer? I think the initial official estimate was 6,000 barrels a day. Does anybody know how you get that estimate? The way you get that estimate is you got to remember that most past oil spills were from ships that ran aground and cracked apart, right? So all the oil was on the surface. So the official Coast Guard way to estimate that is to look at the film on the surface of the water and determine about how wide it is and about what its aerial extent is from a satellite, right? So that's how those 6,000 barrel a day estimates came about, and they were greatly lowballed by BP, right? There was about 1.84 million gallons of dispersant, this emulsifier applied to try to break up the oil into smaller droplets for degradation. This was like a giant experiment. If you consider the Gulf of Mexico as a giant bathtub, essentially they released all this detergent into the bottom of the bathtub with oil and let it swirl around and found out what happened. This was the world's largest accidental oil discharge. Does anybody know what the largest discharge into the marine environment is?
Kuwait.
Yes. When Saddam Hussein released all the oil from the oil production area to stop the coalition forces from invading Kuwait. So that's the largest discharge. This is the largest accidental discharge. So I wanted to show you just a few pictures of the discharge itself. Here you see the rig burning and the ships, the BP ships putting out the fire. This is some footage I got from Ian McDonald that was taken from submersibles, from BP submersibles, and they use this high resolution. I don't know that this is high resolution, but they use high resolution digital video in order to estimate the amount of discharge, the amount of liquid hydrocarbon and gaseous hydrocarbon that's released. It illustrates to you that it's you got to remember this is under incredible pressure, and you have this really hot pressurized liquid and gas mixture that's being released into a cold, dark ocean, right? And so there's an incredible amount of physical chemistry going on as that water hits the deep ocean.
Joe, how big is that pipe?
It's about, I would say that's about as wide as me, probably.
Wow.
As wide as I am tall. I guess. Okay, so I want to show you a few more pictures about, and then just talk about the fate of the oil. A lot of the oil came to the surface and formed slicks. There was a lot of burning of the oil. This is normal. Well, I don't know about normal, but it's normal procedures to respond to the spill. They burn a lot of the oil, and actually this is for the health of the workers that are trying to respond, because there's so much volatile hydrocarbon in there, people get sick. So they have to burn the oil to get rid of the volatiles. And oil doesn't burn so well when it's mixed with water, so often they mix napalm, or they spread napalm on the surface and then burn it that way. So there's a lot going on there, I think, that we don't always know about in the response. This is a cartoon just to show what some of the ways that, well, what happened to the oil. So if you look at the bottom here, you see that broken riser pipe that I showed you video of, and then another pipe was also broken and releasing oil. You can see here that that Corexit, or this emulsifier, was sprayed over the surface of the pipes with, again, submarines. You have to consider that, you know, we can't get down there, right? So everything has to be done by submarine, and that's part of the problem in responding to the spill. That's why it took so long to seal up these leaky pipes. So they spray the Corexit. You can see that that created smaller droplets then that created these large deep plumes that you might have heard about in the media. Then larger droplets rose to the surface and formed these emulsified oil slicks. A good bit of the oil was actually, and I'll show you in a second how much, was pumped up to drill ships, so sucked up directly out of the leaky pipe. There was some skimming from ships on the surface, burning as I showed you. There was a lot of dispersant sprayed from airplanes on the surface, and then some of it evaporated. So this is the official government estimate of what happened to the oil. And if you look in this quarter of the pie chart here, you see that about 3% of it was skimmed, 5% was burned, 17% was extracted by those drilling ships that I was talking about. So about 25% of the oil was accounted for. The other 75% was released into the marine environment, and we don't know really what happened to it. So about 40% of that 75% never reached the surface. We estimate that 10 to 15% evaporated, and 20% was entering the coastal environment, either degraded or buried or is still there. So I'm going to focus on shallow coastal ecosystems, mainly beaches, and I'll show some pictures about the marshes, or of the marshes. This is a picture when oil came ashore to Orange Beach, Alabama, and you can imagine from looking at this that people you know, who run these big tourist operations just want to get rid of it. They want to go away as fast as possible. And actually, they were really successful at putting BP contractors out there just picking it up physically with big machinery. Right, so this oil didn't actually last very long at Orange Beach, but still really affected the economy for a couple of months. Here's a picture of some of the marsh in Louisiana that was affected. You can see that they deployed booms around the fringing marshes to try to capture the oil. It worked to some extent. There was a lot of kind of slopping over the oil into the fringing marsh, so it wasn't perfect. But from my understanding is the oil generally affected the periphery of the marsh and didn't get so much into the interior of the marsh. I put this up here to illustrate kind of the futility of some of the cleanup methods. So here you can see the workers, BP contractors, are trying to clean the blades of the salt marsh grass with baby diapers. They also tried to pressure wash the surface of the marsh. So, and you know, you ecologists and probably anybody can realize if you pressure wash the surface of the marsh, a lot of life in the marsh is right there in the top few centimeters of the benthos, right? So if you pressure wash it, you're going to kill just about everything. So here's a picture of a blue crab that was oiled in the Louisiana marshes. Here's a brown pelican that was affected. You know, it's actually a pretty accurate determination of the severity of a spill is just to see how many birds die. You can actually plot it up, and it's a pretty good statistical comparison. Here you see bottlenose dolphins in a Louisiana sound. Of course, marine mammals will be affected in a variety of ways, including breathing in that material or being affected on their skin. So that gets me to our own research. We've been studying hydrocarbon degradation and the fate of the oil in Florida's beaches. And so that's what I wanted to talk about today. And so I'll start out just talking a little bit about these bacteria. They're ubiquitous in the marine environment. You know, oil is a natural product. It seeps into the deep ocean naturally through these hydrocarbon seeps that you might have heard about. And so a large diversity of microbes, bacteria, fungi have evolved and adapted to eat that great food source, right? A very electron-rich carbon substrate that microbes love to eat. And the second point I want to make here is biodegradation is really the ultimate fate of much of the oil that enters the marine environment. It's either going to be, you know, a good bit of it is just evaporated and physically and chemically weathered, but ultimately biodegradation is very important to the fate of that oil. And so here you see one of the best studied of hydrocarbon degraders, a sunamonad, that says that this oil is delicious. And here you see some emulsified oil particles with bacteria surrounding those oil particles and consuming that oil, or oil droplets. Now, we've been studying the microbiology of shallow coastal ecosystems in the Gulf for more than 10 years. And what we've been focusing on is marine sands and the biofilter that marine sands comprise. And what happens here is that just like the sands in your aquarium, right, a huge amount of water is pumped through these sands, and that exchange of water through the sands, through these permeable environments, stimulates microbial activity, right? So here you see some ripples in the sand, and that topography at the seafloor sets up pressure gradients that pumps hundreds to thousands of liters of water through each square meter of these permeable sands. And what that does is pumps microbial substrates down into the sand, organic matter, oxygen, nutrients, and then it pumps metabolites back out, carbon dioxide and other products of microbial metabolism. We hypothesized then that as oil came ashore, it would be pumped down into the sand and trapped there, clogging this natural biofilter. And that's indeed what happened. So we've been studying the fate of the oil on these sandy beaches. We look at the distribution of the oil and its chemical changes, the response of native microbial communities, and then I'm especially interested in what I'll call the ecophysiology of these hydrocarbon degraders. So this is a look at our experimental approach really for all of the field-oriented projects that are going on in my lab. As I mentioned, we use geochemical techniques to trace microbial activity. We grow these organisms and study their physiology in the lab to predict what they might do in nature. And then we use molecular techniques to look at their community composition, their abundance, distribution, their ecology in general. So we use quantitative PCR to look at abundance. We do pyrosequencing of amplicons, as Frank was talking some about yesterday, and then metagenome sequencing as well. So what I wanted to do next is take you through a series of slides of what happened to oil on Pensacola Beach, basically. So we believe that oil began to come ashore in early June, and what happened was it was deposited in this like 30-meter wide swath. It's very hot in Florida in June, so the oil heats up and pools in depressions on the surface of the beach. But beaches are really dynamic environments, right? The sands are constantly shifting, depositing, accreting, eroding. And so within a week's time here, you can see that this oil already starts to get buried in the sand. So it's not only that pumping effect I was talking about, it's just the deposition of new sand. About a month after that, then, you see that the oil has already been buried over 50 centimeters in the sand column here. You can see clean sand on the top, and what happens is as clean sand is deposited on these oiled layers, it acts to squeeze the oil out into the surrounding pore waters, and it kind of seals off, physically seals off those layers, affecting their biogeochemistry. By September or October, though, it's a relatively good story in beaches. We saw that already these layers started to degrade, started to fade from weathering biodegradation. Okay, so here you see, and I should mention Will Overholt, the data that I'm going to show today. Will is a new PhD student in our department. John Delgardio is my longtime technician, and here you see him collecting samples for microbial community analysis. So the layers already started to fade by September, October. It was decided in October, on October 20th, that BP contractors would go in and try to clean up these oiled layers. Okay, so they brought in these big machines. This is just a physical cleaning. So they dig up the sand, they pulverize it, sieve it, mix it with clean sand, and then dump it back onto the beach. Okay. And so here you see them doing that. The question is, was the beach really clean? Here you see an image that was taken in December, shortly thereafter. And what you can see is the top couple of feet of the beach is still brown, stained with oil, so the oil did not go away. But what it did act was to spread out that oil over large areas of the beach, right? And one could argue too about other unintended consequences, such as when you pulverize and sieve all the sand, you probably kill all the invertebrates in there as well. No one asked us what to do about this. No one asked any of the scientists. The people that were working on these beaches just said, we got to get it cleaned up because we want people to come to our condos, and so they just cleaned it up. So here we're studying an inventory of the oil hydrocarbons that were in these sands, plotted with time. So here you see buried oil with time. And like I said, this was a good story. Much of the oil that could be degraded was degraded within just the first 4 months. Okay, and this was weathered oil. You got to think it's released from the pipe, transported all the way through the ocean water column, and eventually deposited on the beach. So even the weathered oil apparently is a good substrate for microorganisms and is biodegraded. Right. We also follow the activity of these microorganisms by measuring just simple oxygen consumption. Okay, and you can see that the blue dot shows oxygen consumption in a pristine sand, the red dots in oiled or contaminated sands. So you can see we got 2 to 4 times more oxygen consumption in the oiled sands associated with hydrocarbon degradation. That also went away within the first couple of months. And then post deep cleaning, we really didn't see too much difference. There remains a somewhat elevated oxygen consumption after that time. Now more about the microbiology. And as I said, we grow these organisms in the lab, and then we also study their response with molecular techniques. One of the important parts of our response anyway was to be able to get our hands on some of that oil. It was a very sensitive issue at that time. The BP didn't really want to let that oil get out. And so I was lucky enough to have a colleague on one of the drilling ships that took a lot of that oil for me. And so we were using that to enrich these hydrocarbon degraders and study them while we were taking field samples. So that was an important part of our work. And here you see an aerobic enrichment of oil degraders from these contaminated sands. So what we found basically from our cultivation studies, here you see some more of these aerobic enrichments, is basically that under ideal conditions in the lab, oil would be degraded in a matter of days, aerobically. Okay? When we enriched these organisms anaerobically with sulfate or nitrate as the electron acceptor, it took a long time, if the oil was degraded at all. Right? We had some of these enrichments years after. We could smell a little bit of sulfide in the enrichment from sulfate respiration, but hardly any of the oil was even used in the culture. So, point is, oxygen is very important to degradation. Degradation will occur under anaerobic conditions, but it's going to be very slow. And we can talk about what that means for ecosystems. So we also then isolated some of these hydrocarbon degrading bacteria. Here you see 24 strains that we've published on. Many of them are members of the gamma proteobacteria that have been detected in oil contaminated marine environments before. And you can see just from their names that they're usual suspects. Alkanovorax dzeolii, which eats diesel fuel, and Marinobacter hydrocarbonoclasticus, which eats oil as well. We also found some unusual, unusual organisms as well. Some members of the alpha proteobacteria that haven't been studied too much, and the members of the actinobacteria that we think are important. We think that the gamma proteobacteria bloom up first and eat a lot of the straight chain compounds, the aliphatics and the alkanes, and then you have other organisms coming in like these alpha proteobacteria and these gram-positives that are eating some of the more branched alkanes and the more recalcitrant aromatic compounds. So the first thing we did was then test these isolates in the lab just to show that they degraded oil and they grow on it. That's what you see here. I actually had an undergraduate student at Georgia Tech that got a Pura fellowship this summer to do that work. And we've been also working with Terry's lab to understand how toxicity is related to biodegradation. I'm not going to show those results today, but it actually turns out to be very interesting. Now here is every microbial ecologist at some point shows an unreadable phylogenetic tree in their presentation, so now I have to have one too. So here you see the unreadable phylogenetic tree, and all it shows basically is what we could grow is closely related to what we retrieve from the environment in terms of the gene sequences. So this is a phylogenetic tree of the small subunit ribosomal RNA gene sequences. The stars indicate our isolates, and what I want to show you here is one group in particular, the alkanovorax group. This is one of the best studied of hydrocarbon degraders under aerobic conditions anyway. It's well known to degrade alkanes in particular, that's why it's called alkanovorax. But what I want to show you here is that the stars, our isolates, were highly related to the sequences that we retrieve from the environment. So we think, at least in this case, we have some pretty good model organisms to use to study the physiology of these bacteria. We also, this is pretty much the same thing we did in the larger flasks. We just control the enrichment to try to enumerate the hydrocarbon degraders. We saw that in the oiled sands, there were orders of magnitude higher numbers of hydrocarbon degraders than we got in clean sand. And then if we follow that up with molecular techniques, here I show from quantitative PCR the relative abundance of overall bacteria, and then also the hydrocarbon degraders I was talking about, the alkanovorax. We designed primers for those hydrocarbon degraders. And what you see here is the gene abundance with time in 2010. You can see that there was an abundance and a bloom of alkanovorax bacteria initially in the first couple of months. We see then a bloom of overall bacteria. The deep cleaning event happened here. After deep cleaning, we see another stimulation of these hydrocarbon degraders after the particles were all mixed up again, and the abundance remained low after that. And you can see too that the abundance correlated with the presence of oil. I wanted to talk briefly also about the composition and diversity of the microbes that we find that are eating the oil. Here we've looked at profiles across the zone of oiled sediments. We've sampled with cut-off syringes across that oil layer. And we use 454 pyrosequencing of the small subunit ribosomal RNA gene sequences to study the community composition in the various layers. So here you see the core. We study layers above where the oil is, and then below. And you can see in the oiled layer, we see a large enrichment of proteobacteria, the gamma proteobacteria that I mentioned that eat the oil. But you can see it's an amazing, almost a pure culture of these hydrocarbon degraders. So 50% of the community in relative abundance anyway, sequence abundance, was Marinobacter. Another 11% was Idiomarina, both of which are gamma proteobacteria that are known to degrade oil. So one of the things we're doing now is we have 10 metagenomes from these depths, and now we think it's a great opportunity to try to construct genomes de novo from these samples. Here we've taken 30 to 40 pyrosequence libraries from the DNA that we extract from these sands, and also the RNA that we extract from these sands, and do a reverse transcription reaction. The size of the bubble is proportional to the relative abundance of alkanovorax in the libraries. And then here it's an MDS plot of the Bray-Curtis similarity index taken from the pyrosequence data, where the distance between these values is basically phylogenetic relatedness between the communities. And so what you can see is the pristine samples clustered together on this side of the graph, and then you see a progression or a pronounced shift in microbial community composition caused by the oil, both at the DNA level and also at the RNA level. So that leads me to the end. How am I doing on time?
Great. A minute till my time.
Oh my gosh. Okay. So I wanted to convey to you that the Gulf is a vast, beautiful, and important ecologically and economically. That oil and gas production is not going to go away. Okay, 75% of the oil we did not really adequately account for that was released from the Deepwater Horizon discharge. The beach story is good. The oil was largely biodegraded. The salt marshes that were affected is not such a good story. The oil remains there, at least at the fringing part of the marsh, and will be degraded much more slowly in these highly impermeable sediments. We see, I didn't really present all the data, but we see a succession of oil degraders in these oiled sands, as I was alluding to, from the gamma proteobacteria, well, within the gamma proteobacteria, and then with other groups. And also we see with the data that I didn't show together with Terry that biodegradation leads to a decrease in the toxicity of the oil and corexit. Thank you.
Something that you find about later, and it's kind of like, well, that was stupid.
Well, you notify BP that it's stupid? I don't know what the other... They were doing a lot of stupid things too. But no, I see what you're saying. I mean, there certainly was, and scientists are to blame as well. There was a real lack of coordination amongst all the stakeholders. The government was in charge. The Coast Guard was in charge of response efforts, but that was largely out at the well site. Right? What was going on in the coast was not controlled. It was largely controlled at the municipal level, and I think that's why really there was no coordination. Those people were saying, "I want that beach clean. Let's clean it up now." So they would call BP, and BP would send in the bucks. And some of this, I didn't even talk about half of it. And around Florida, there were areas that were never affected by oil where they had huge response. People just going out just to check for oil, you know. And actually really disturbing the ecosystem more than anything else. Yeah?
So since 75% of the oil left in the environment, you just told us the story about degradation, so it's the 75 that wasn't sort of active?
No, that would be included. The degradation would be included in the 75%. I say that to just talk about the lack of knowledge of the fate of the oil, but I imagine much of that was biodegraded. The tough part was that the oil that's in the deep ocean is really hard to track, right? We can say what the concentration of oil was in these deep plumes. We can map where those deep plumes went. There have been models already developed to show where that oil goes, but how do you really account for all that oil? I mean, you can model it, you can measure it, but a lot of it you just don't know where it goes.
Do you worry, at least somewhat paradoxically, that the results saying that microbes taking care of these can degrade some portion of the oil will somehow lead to BP or some future, let's say, driller saying, well, look, you know, if it spills, no big deal.
It's yes, of course, and of course I tell the good story, right? Because it's something nice to talk about. But there are a lot of bad stories out there too. In fact, I didn't really get to show when I presented this at the Georgia Aquarium. I show a lot of examples of how the deep ocean habitats have been affected. And so there's examples now about how the deep corals have perished in response to these oil plumes flowing over the top of them. So yeah, that's our job, is to convey our results, and then the powers that be make those decisions. But that's a big part of what we're doing in our program, and we hope to be funded 10 years to do this. So we'll see. Yeah, Kirill.
What would be your smart way?
Pardon me?
What would be the smart way or your way to deal
Yeah.
with the situation?
For the beach.
To understand correctly, just dump instead of soap, bacteria.
Yeah.
Do they work?
So I will talk about the beach because that's what I know, that's what I've been working on, and I think as much oil as you can collect with booms. We've learned a lot about booms just from this response effort, and I think we can build better booms. So I would collect as much of it as we can. I think that's not, and then you can protect sensitive areas that way. For example, national seashore habitat that's along the Gulf. And then I would just leave it there. I don't think that the oil had an adverse effect, at least on beaches, on the surrounding ecosystem. So I would not use the deep, I don't think the deep cleaning had a very important effect on the remediation. And this was also borne out in the response to the Exxon Valdez spill. You know, what they did for the Exxon Valdez, also a lot of beach and coastal environment is they fertilized the whole area. And that sped up the degradation, and I think that was also the intent of deep cleaning, but neither effort really changed the extent of degradation, you know, a couple of years later. In the case of Exxon Valdez, 5 to 10 years later, in my opinion. Yeah.
What is the, is it me?
Yes, sir.
Yeah, okay. So, using the dispersants, do we know what that really did? And is it a good thing to get it in small droplets so you can get a lot of degradation quickly? Or, you know, was the dispersant worse than the initial problem, and do we know?
I don't know that we know. Terry studied the toxicity some. He can tell you that Corexit makes oil a lot more toxic. He and Roberto found 52 times more toxic than the oil or the Corexit on its own when they're mixed together in ratios that the EPA recommends. Right? Now, we haven't studied that a lot in our cultures. We're just starting to study that now. Did it help? I think it did help. And I think it helps because if you break up those droplets in terms of biodegradation, not in terms of ecological impact, but it breaks it up into smaller droplets. You think about the deep ocean. What do oil degraders need, right? They need a food source, which is the oil. They need oxygen, which is the process is much more rapid. And they need major nutrients. The deep ocean is rich in major nutrients, right? And there's plenty of oxygen. So you optimize the biodegradation in the deep ocean by adding Corexit. Now, as far as those unintended consequences go, then I'm not sure about that. The toxicity, I think, is still, there's a lot of data in the NERDA process, the National Resources Damage Assessment, that we haven't seen. I think there's a lot of information on toxicity yet to come out. The stuff, from what I hear, doesn't biodegrade very fast, though. So. The work that I've seen by Terry Hazen and others, when they did biodegradation studies just in enrichment cultures, they get really slow rates. So, yeah?
One more question.